Good morning, students. Today we will study structure of nucleic acids. In the last lecture, we had studied the structure of nucleoside and nucleotide. Nucleic acids, that is DNA and RNA, are the polymers of nucleotide, and they are formed by joining third prime position OH group of one nucleotide with fifth prime position sulfate of other nucleotide, and that is shown in the figure 14.28. This we will see. This is figure 14.28. So this is DCMP. We know this long form very well now. D stands for 2-deoxy or deoxy. C for cytosine. MP for monophosphate. So this is a ribose sugar, do not containing OH group at second prime position. This is base present, that is cytosine, and fifth prime position contains the phosphate group. So this is example of nucleotide. This nucleotide plus DAMP, that is deoxy adenosine monophosphate. So this is a sugar, do not containing OH group at second position. Here the base present is adenosine and the phosphate group is present at fifth prime position. Here these two nucleotides get joined. The fifth prime position of this DAMP nucleotide get bonded with third prime position OH group of other nucleotide, and this bond is formed, that is called as a phosphodiester linkage. Phosphodiester linkage, and this is a formation of dinucleotide. Like this, many nucleotides join together to form a polynucleotide, and that is RNA or DNA, that depends upon the second position OH group present or not. So this phosphate group is present of DCMP nucleotide. Here the fifth end is a free. Fifth prime carbon, that end bonded with phosphate group, this is a free end. And here in other nucleotide, that is DAMP, here third position OH group is a free. So it is a third prime position end and it is a fifth prime position end. These two nucleotides are joined together by phosphodiester linkage. So like this, the two ends of the polynucleotide chain, they are distinct from each other. One end having the phosphate group at fifth position called as a 5 prime position end, and other end is third prime position as a free OH group at third prime position. This we had studied in this structure. One nucleotide have 5 prime position free end and other nucleotide have 3 prime position OH group is at the free end. The primary structure of nucleic acid is the sequence of the nucleotides in it. So in the primary structure, we will observe mainly this sequence of nucleotides, in which sequence these nucleotides are joined together. This in turn determines the identity of the base in the nucleotides. The different nucleic acids have distinct primary structure. It is the sequence of bases in DNA which carries the genetic information of organism. This is the main function of DNA. The sequence of bases in DNA which carries the genetic information of the organism. The polynucleotide chain of nucleic acid are named by sequence of bases. The beginning at fifth prime position end using one letter symbol of the base. For example, here this example is given, that is name CATG, means there are 4 nucleotides in the segment containing base cytosine, adenine, thymine and guanine in the indicated order from the fifth end. So this structure, that is the schematic representation of the nucleotide segment ACT of DNA molecule. Here the fifth prime position at one end and third prime position at other end. The phosphate group, then deoxyribose, this A is adenine, then phosphate deoxyribose, then cytosine, phosphate deoxyribose, then thymine at 3 prime position. This is the nucleotide segment. Then the DNA double helix molecule. So James Watson and the Francis Crick, called as a Watson and Crick model, they had given this model in 1953. This model is a double helix model of DNA structure, which has later verified by electron microscopy, and the salient features of the DNA model, that is a double helix structure, it is shown in this figure. So these salient features are, first is DNA consists of 2 polynucleotide strands that wind into right-handed double helix. So this is a 5' end, and this is a 5th prime end, free ends. This is a one strand that is 3 prime position end containing, and other end is a 5 prime position. Other strand have 3 prime position, free end, and it is ended with 5 prime position. This dotted line at the center, that indicates the axis of helix. On this strip, on this strand, there is a sugar phosphate backbone, and in between these AT, CG, this TA, CG, these are the bases, and these bases are joined together with the help of hydrogen bond. This light blue color base is a adenine, dark blue colored is guanine, pink colored is cytosine, and gray colored is thymine. So all these bases are shown with their particular colors. Second important salient feature is 2 strands run in opposite direction. Just now I had explained that for one strand there is a 5th prime position at the top and 3rd at the bottom, and opposite for the other strand. So they are oppositely placed. Then the sugar phosphate backbone lies on the outside of the helix, and the bases lies on the inside perpendicular to the axis of helix. These are the bases, they are perpendicular to the axis of helix. Then the double helix is stabilized by hydrogen bonding between the bases. These dotted lines, or in this structure, these dotted lines that shows the hydrogen bond between the bases. And due to that, this double helix structure is more stable. So this gives rise to the ladder-like structure of DNA double helix. Adenine always forms the 2 hydrogen bonds with thymine, and guanine always forms the 3 hydrogen bonds with cytosine. Therefore, there are adenine, thymine, and cytosine, guanine. These bonds are complementary bonds. The base pair and the two-strand double helix are complementary to each other. It may be noted that the RNA exists as a single-stranded structure. Double stranded structure is for DNA, and single-stranded structure for RNA molecule. And that is about the structure of DNA and RNA. Thank you.